Okay, hello everyone. So welcome to our second to the last topic for our Biology 100, which is on the nervous system. So recall that one of our first lessons in Biology 100 was on the attributes of life. And one of these attributes is irritability, or the ability to detect and respond to external stimuli from our environment. So this is where the nervous system actually comes in. So to guide us, allow me to show you the timeline, the outline, I should say, for this particular lecture. First, we shall be discussing the fundamental units of our nervous system, which are the neurons or the nerve cells. So much of the structure of these cells have already been discussed during your histology lecture. So I will no longer be dwelling on these. So instead, we shall be discussing nerve signal transmission or the mechanisms of how these neurons communicate with each other. We shall also be discussing the other cells present in the nervous system, or the glial cells or neuroglia. Then finally, we shall be talking about the central nervous system and the peripheral nervous system. So note that we will be focusing on the human nervous system, which is an example of the bilateral nervous system. So remember in your lecture in histology, it was also mentioned that there are different types of nervous systems. The simplest can be seen in your animals with radial symmetry, such as your hydra or jellyfish or other cnidarians. So these organisms have a nerve net, which is simply a network of connected neurons. So for more complex organisms, such as humans, we have bilateral symmetry. That is, given our sagittal plane, the left side and right side are mirror images. And because we have bilateral symmetry, we have our bilateral nervous system that developed from cephalization and centralization, allowing for a more defined central nervous system. So for this lecture, again, we will only be focusing on the bilateral nervous system, which is exhibited by humans, while other types of nervous systems will be discussed in your other zoology courses, such as your Zoology 102 and your Zoology 111. Right? So let's start. So you know that the fundamental units of our nervous system are the neurons or the nerve cells. So again, the structure and important parts of these have been discussed in your histology lecture, so you may just go back to that lecture. But for this slide, I just want to emphasize these three types of neurons that have their own specialized function, important for nerve transmission. So one, we have our sensory or afferent neurons that convey information from your sensory receptors to your central nervous system. Okay, sensory receptors to CNS, so that is your sensory or afferent neurons. Okay, the second one, we have our motor neurons. Let's go to the motor neurons first. And these motor neurons convey information from your central nervous system to your effector cells. So what are your effector cells? Essentially, your effector cells are, these can be your muscles or your glands, etc. And third, we have our interneurons. So our interneurons connect our sensory and motor neurons. And in most animals, this is located entirely in the central nervous system. Okay, so this figure in the right, kindly look at this figure in the right. This illustrates the knee-jerk reflex. So I'm sure we're all familiar with this reflex. So basically, when our knee is tapped, we kick. So what happens here is that we have our sensory receptors in our sensory neurons. These sensory neurons here, detect the sensation, this stretch in the muscle essentially, and converts this stimuli, this environmental stimuli, into nerve impulses. So these nerve impulses will travel down the sensory neuron, travel down the sensory neuron, this blue neuron, into the central nervous system. And since this is a reflex, this no longer goes to the brain. It is already integrated and analyzed in the spinal cord. So that's what's interesting about reflexes. These no longer have to be integrated in the brain anymore. And from the central nervous system, particularly the spinal cord, this information is conveyed down to your motor neuron, here, this purple neuron, and then to your effector cells, which in this case is your muscle. So it is noticed in this figure that interneurons can either be present or absent between the sensory and motor neurons. So they can either be present, this green neuron here is an interneuron, or they can be absent between your sensory and motor neurons. So just nice to know, for sensory neurons that connect directly to motor neurons, these are called monosynaptic, while sensory neurons that are connected to motor neurons through interneurons are called polysynaptic. Okay. So from our sensory receptor to our sensory neuron to our central nervous system, to our motor neuron, and to our effector cell. This is the basic pathway from detecting a stimulus to a response. So in this example, our effector cells, again, are the muscles, particularly the quadriceps. So these quadriceps, they contract, and the flexor muscles you have your quadriceps and you have your flexor muscles. So in the knee-jerk reflex, your quadriceps, they contract while your flexor muscles, they relax. Thus, this action actually allows us to kick when our knee is tapped. So that is what happens in this knee-jerk reflex. All right. So to better understand how this works or how nerve signals are transmitted, we need to know the concept of a membrane potential. So a membrane potential is basically the difference in electrical charges or the difference in the electrical potential of the inside of the neuron and the outside of the neuron. So here we say that the inside is negatively charged relative to the outside, but this happens in the resting state or when the neuron is essentially resting. Okay, so here let's consider a resting neuron or a neuron that is not transmitting a signal. So the interstitial fluid surrounding the neurons, so here in the outside, they contain relatively high concentrations of sodium and chloride here. So this is the interstitial fluid outside of the neurons. So they contain relatively higher concentrations of sodium and chloride ions, but they have a low concentration of potassium ions and also low or almost zero concentration of large anions. So these large anions are impermeable, so they cannot pass through the cell membrane. Now, this is what happens in the outside, but in the inside of the neuron, we can see that this is actually reversed. So inside the neuron, we have a high concentration of potassium ions and large and impermeable ions, but we have a lower concentration of our sodium ions and our chloride ions. So essentially, the inside of the neuron is more negatively charged. It is more negatively charged compared to the outside of the neuron. So inside is more negatively charged compared to the outside due to this unequal distribution of these ions. And it is important to note that the resting membrane potential is about -70 millivolts. Okay. All right. So ito yung sinasabi ko kanina. I'm so sorry about that. Now, what is important to know as well is that at resting potential, there are open or ungated channels that allow the diffusion or leakage of ions. So there are open channels even in our resting neuron that allows the movement of particular ions inside and outside of the cell, particularly following the gradient. So we have our potassium and sodium channels. And it is important again to note that we have more potassium channels that are open compared to sodium channels. So basically, more potassium leak towards the outside of the cell. This makes the inside of the cell or the inside of the neuron even more negative. Okay, so we have more potassium channels than sodium channels. So more potassium channels that can get out of the cell, making the inside of the cell even more negative. So to be able to maintain the resting potential, which is at negative 70 millivolts, again, we have this sodium-potassium pump. And this sodium-potassium pump actively, so when we say actively, they utilize ATP. Recall that lecture on membrane transport. So they actively transport sodium ions out of the cell and potassium ions into the cell. All right. So this again maintains the resting potential. Okay. So this is what happens in a resting neuron. Okay. So this figure in this next slide summarizes the transmission of nerve signals. So let's see first this first step. This is step 1. Here we have the cell that is at resting potential, again at around negative 70 millivolts. So it is important to note that the cell membrane of our neuron actually contains many types of channels. You have your open channels, your always open channels, which I discussed earlier, and your sodium-potassium pump, which helps maintain your resting membrane potential. Now, apart from these channels, we also have your voltage-gated sodium and potassium channels that are closed. And these are actually sensitive to changes in voltage. Okay, so recall that lecture on membrane transport. But again, at resting membrane potential, these particular voltage-gated channels are closed. Okay, remember that. So what happens if a stimulus is applied? So a stimulus can be anything. It can be light, touch, sound, etc. So anything that can be received by your sense organs is your stimulus. Now, when a stimulus is applied, this opens a few sodium ion voltage gated channels, thus allowing sodium ions to enter the cell. Now, the strength of the stimulus actually plays a part in this step, because if the stimulus isn't strong enough, then only few sodium ion voltage gated channels are open. Thus, only a small amount of sodium ions enter the cell, making the inside of the cell slightly less negative. All right. But for an action potential to occur, the voltage inside of the cell should actually reach a certain threshold, which in this example is at around negative 50 millivolts. Ito yung threshold sa example na ito. So again, if the stimulus isn't strong enough, it will only be able to open a few sodium ion voltage gated channels, which only allows a few sodium ions to come in, making it only change the voltage very slightly. But if the stimulus is strong enough, this would actually open more sodium voltage gated channels. And this would then allow more sodium ions to enter, making the inside of the cell less and less negative, possibly reaching the threshold. So this is an all or nothing process. Either an action potential occurs or not at all. Either enough yung sodium ions that entered the cell or hindi. Okay? So this is what happens when a stimulus is applied. It's whether a threshold is reached or not. Now, what happens if the threshold isn't reached? If the threshold isn't reached, then our neuron goes back to the resting potential. So no nerve signal will be transmitted. But if the inside of the cell reaches that threshold of negative 50 millivolts because of the influx of sodium ions, then an action potential will be triggered. So when the threshold has already been reached, then this will open even more sodium voltage gated channels. This will open even more sodium ion voltage gated channels, thus allowing even more sodium to enter the cell, making the inside of the cell even more positive very abruptly. All right, ito yun. Itong part yan ng graph. So basically, this is now what we call your depolarization phase, because the interior of the cell is now more positive than the outside. So the polarity reverses quite abruptly. All right, that is your step 3. And then once it reaches your peak voltage, so our peak voltage here is at around 30 millivolts. So this would now trigger the sodium voltage gated channels to close. So once this peak has been reached, this will trigger the closing of our sodium ion voltage gated channels, and they will now become inactivated. So hindi na pwedeng pumasok ang sodium ions. So now what happens is that the potassium ions will now open. The potassium ion voltage gated channels will now open, allowing your potassium ions to exit the cell. So this makes the inside of the cell more and more negative again, kasi our potassium ions, which are positively charged, are leaving the cell, thus making the inside of the cell more negative. Ito yun. And thus we call this our repolarization phase. So depolarization ang step 3, while repolarization ang step 4. Now it is important to note that these potassium voltage gated channels are very slow. Relatively slow sila sa pag-close. Mabagal silang mag-close. So this makes the inside of the cell even more negative, and it actually undershoots. Ito undershoots the normal resting potential very briefly. And this is known as hyperpolarization. So the voltage inside the cell very briefly is even more negative than that of the resting membrane potential. But this happens very briefly. And then the neuron recovers and it goes back to its resting potential, which is our step 1. All right. So that is what happens. This is what happens in the transmission of nerve signals. Okay, it's a very brief summary. Now we have to understand that an action potential, I should say, it actually happens in a very specific place in a neuron. It is localized. Okay, so it is localized. And what happens now is that it has to be passed on from the part of the axon which is near the cell body down to the synaptic terminals, which can be found at the end of the axon. So how can a localized action potential be transferred down to the ends of the axon? So this actually is one property of our nerve impulse. It is self-propagating. So the action potential propagates itself along the axon. Okay, so let's discuss that. So we see here in this figure, let's focus on this figure. here in step one, there is an inflowing of sodium ions from the first action potential in this particular localized region. Okay, so there's an influx of sodium ions. So recall lang kung bakit yan. And then in this region, the spread of the electrical changes caused by this influx of sodium ions is actually enough for the neighboring region, for this region, let's just say, to reach threshold in itself. Okay, so here, when sodium ions flow in due to the action potential, the spread of this is actually enough to trigger this neighboring regions to actually reach threshold. And thus, here in the neighboring regions, this will actually trigger an action potential because it has reached threshold from the influx of sodium ions from this first region. Okay? And we have to note that this happens along the axon up to the synaptic terminals. So if your action potential starts here, again, the influx of sodium ions is strong enough to make the neighboring region reach threshold and thus triggering a new action potential, which is this one. Okay, and this will, again, this influx of sodium ions from the action potential that was triggered is strong enough to make this neighboring region reach threshold, thus triggering another action potential here. Okay? So this happens along the axon up to the synaptic terminals. And we have to note that it only moves towards the synaptic terminals and not back to the cell body. So one way yung movement nila. And basically, this is because that, recall that during the repolarization phase, the sodium-gated channels become inactivated. Na-inactivate yung mga sodium-gated channels dito. All right? Once there is already the repolarization phase. So na-inactivate yung nagko-close or na-inactivate na yung sodium ion voltage gated channels natin dito. Thus, hindi na pwedeng pumasok ang sodium ions. So, hindi pwedeng mag-trigger ng action potential in this region of the axon anymore. Kaya one way lang ang propagation ng action potential. Now, the action potential can happen or can be propagated in different ways. So one is through continuous conduction, and this occurs when the neuron is unmyelinated. All right. So this unmyelinated neuron is present in some invertebrates. So what happens in continuous conduction is that the action potential spreads continuously, walang interruptions, walang gaps. And this is actually quite slow. Mabagal ito kasi it will pass through this entire surface. It will spark or it will trigger action potentials in this entire surface. So this is quite slow. And this is quite slow compared to our saltatory conduction because the polarization, it happens everywhere. Okay, it happens along the entire surface of your axon. While in saltatory conduction, such as in what we experience in vertebrates, when the neuron is myelinated, as you can see here, so this is present in vertebrates, the myelin sheath is actually interrupted. All right, ito yung myelin sheath natin. And you can see that there are some gaps at the interval. So these are interrupted in a way that the axon here, this is our axon, this purple one. So in this particular nodes, the axon is exposed, while here, where it is surrounded by the myelin sheath, it is not. Now, these nodes where the axon is exposed are called your nodes of Ranvier. So this myelin sheath is to prevent the polarization in parts of the axon that it surrounds or covers. So what happens is that the action potential actually leaps from node to node. And it is in these nodes where the ion channels and pumps are concentrated. So once an action potential starts here at our first node in our axon hillock, sodium ions enter and creates this force that pushes your ions inside into the next node. And once this next node, the ions reach this next node, it is enough to actually reach threshold, thus triggering another action potential. All right. So saltatory conduction, there is this leaping from node to node, and it is much faster than continuous conduction and is more efficient. So now what happens when the signals reach the end of the axon? So these are now passed to the next cell or the receiver cell, and these happens at the synapse or relay point. So the next cell or the receiving cell can either be another neuron or an effector cell. So these effector cells can either be your muscles or your glands. All right? And synapses can either be electrical or chemical. So electrical synapses are found in the heart and digestive tract in humans. And these are fast as the two cells, your sending cell and your receiving cell, are essentially connected through gap junctions. So in this type of synapse, the signal flows directly from neuron to effector. There is no gap. While in chemical synapses, which is what we can see here in this illustration, there is a narrow gap between the two cells. And this narrow gap is known as the synaptic cleft. So the signal cannot flow directly like the electrical synapse. So what happens is that the action potential is converted to a chemical signal in the form of neurotransmitters. So yung neurotransmitters natin ay ito, yung mga brown na bilog na yan. So when the action potential reaches the synaptic terminal, this will now cause these vesicles, itong mga vesicles dito, that contain the neurotransmitters. So we call these vesicles that contain neurotransmitters your synaptic vesicles. So this will cause the synaptic vesicles to fuse with the plasma membrane of your sending cell. And then through exocytosis, these vesicles will release your neurotransmitters into the gap or into the synaptic cleft. All right, ito yun. So through exocytosis, the vesicles release your neurotransmitters into our synaptic cleft. So in the receiving cell, here, the receiving cell, ito yun. This is the cell membrane of your receiving cell. There are ion channels, again, in our plasma membrane. And these are sodium ion channels or potassium ion channels, etc. So what happens is that the neurotransmitters will now actually bind. They will bind to the receptors of these channels. And this will now open the channels where ions will enter the receiving cell and start new action potentials, especially if they are excitatory. So makikita natin mamaya that neurotransmitters may actually bind with receptors of different channels, thus making the effect either excitatory or inhibitory, which we will see later on. Okay, so if it will bind to, let's just say, a sodium ion channel, then this will allow sodium ions to enter, thus triggering an action potential. Now, what happens is that this actually happens quite briefly, and an enzyme will now break down your neurotransmitter. And once the neurotransmitter is broken down, this will now close the ion channels. Okay? So, once the ion channel is closed, syempre, hindi na pwedeng pumasok yung mga ions. Okay. So, basically, one-way action lang ito. From sending to receiving. Hindi siya pwedeng pabalik. Alright? So, in this slide, sorry about that. Here in this illustration, we can see that a single neuron may actually receive information from many different neurons. Alright? So this is the cell body. You have your dendrites here, and you can see that this single neuron can actually receive information from many different neurons. You have a neuron here, you have another neuron here, you have another neuron here. Okay, and these different neurons may actually send varied information to the receiving neuron. So neurotransmitters released by the sending cell, they may either bind to sodium ion channels or to potassium ion channels. So if the neurotransmitters bind to sodium ion channels, they allow sodium ions to enter the receiving cell, thus triggering an action potential. Okay, because the inside of the cell becomes more and more positive, thus reaching a threshold, or the possibility of reaching threshold, and thus triggering an action potential. Now, the effects of these neurotransmitters that open sodium ion channels are excitatory. If the neurotransmitters, however, bind to ion channels that allow potassium to exit the cell or chloride ions to enter, so this actually decreases the tendency of an action potential. Why? Because the inside of the cell becomes more negative. So if the inside of the cell becomes more negative, this decreases the tendency of an action potential. Thus, the effects of these are inhibitory. And again, this receiving cell can receive a mixture of excitatory and inhibitory signals. So an action potential will be generated in our receiving cell if the excitatory signals are collectively strong enough. And this would make the inside of the cell more positive. So this would raise the membrane potential to threshold and trigger an action potential. And then it will, this action potential will be passed to its own axon and then to the next receiving cell. Okay? So dapat collectively strong enough yung excitatory signals. Okay. And just nice to know, acetylcholine is a neurotransmitter that has both excitatory and inhibitory effects. So this is excitatory at skeletal muscles, in which it makes the skeletal muscles contract, but inhibitory at the cardiac muscles, in which it slows the rate of the contraction of the heart muscles. Okay. So now we go to the glial cells or neuroglia, which are other cells apart from the neurons that are present in the nervous system. So the glial cells found in the central nervous system are the following. Our astrocytes, which provide for nutrition and structural support. Our ependymal cells, which line our ventricles. So our ventricles in our brain are cavities that are filled with cerebrospinal fluid. We also have our oligodendrocytes, which form the myelin sheath. So these are the cells that form the myelin sheath that surrounds part of our axons and the neurons of our central nervous system. And finally, we have our microglial cells, which are essentially macrophages and involved in immune defense. The glial cells found in the peripheral nervous system are the satellite cells and the Schwann cells. So the satellite cells, just like the astrocytes of the central nervous system, provide for nutrition and structural support. The Schwann cells, on the other hand, they form the myelin sheath that surrounds the axon of the neurons of the peripheral nervous system, just like the oligodendrocytes for the CNS. Okay. So the cells that form the myelin sheath for the CNS and the PNS are different. Okay. So these are the other cells present in our nervous system apart from the neurons. So now we move forward to the bilateral nervous system as exhibited by humans. So again, humans have the central nervous system and the peripheral nervous system. So the central nervous system is basically composed of the brain, which is the control center. And the brain is the control center and the spinal cord. So these are the parts of our central nervous system. So seen in this illustration are some parts of the brain and the spinal cord. We have the ventricles here. Again, which are cavities that contain the cerebrospinal fluid, and this is continuous with that of the central canal and the spinal cord. So the cerebrospinal fluid that circulates throughout the central canal, itong central canal ng spinal cord natin, and ventricles, they actually cushion our central nervous system and assist in supplying nutrients. Next, we have the meninges. So here, let's just zoom in. Our meninges are these gray layers of connective tissue that surround the brain and the spinal cord, which also serve as protection. So cerebrospinal fluid also circulate between the meninges, adding protection to the CNS. So in between these two layers of meninges, there is the cerebrospinal fluid. Same is true here in the spinal cord. Now, the blood-brain barrier, not shown here in the picture, is also another mechanism that protects the brain. So this is due to brain capillaries that allow only the essentials like oxygen to pass into the brain. But this blood-brain barrier, it keeps out many other chemicals. Okay. And the other structures that protect our central nervous system, apart from the meninges, the cerebrospinal fluid, and the blood-brain barrier, is our bone structures. So we have our skull, which protects the brain, and our spinal column, which protects our spinal cord. So in our spinal cord, which is simply a bundle of nerve fibers, we have our gray matter here and our white matter. So our white matter is composed of axons, while our gray matter is composed of dendrites and cell bodies. Okay, so now let's focus more on the brain. So here we see the brain and its different parts as seen from the side and from the back. So now these different parts of the brain are specialized from 3 bulges that appear at early embryonic development. You have your forebrain, your midbrain, and your hindbrain. So our cerebrum, our thalamus, and hypothalamus, they developed from our forebrain. Our midbrain, obviously from the midbrain in the early embryo, and our cerebellum, pons, and medulla oblongata are developed from the hindbrain. So here you can see the functions of each of these parts of the brain, and you can just go through these functions yourself. You will be learning more of this as you go into your animal physiology. So now let's go to the peripheral nervous system. So essentially, our peripheral nervous system consists of nerves and glial cells that lie outside of our brain and our spinal cord. So our peripheral nervous system has two functional divisions. You have your sensory division and your motor division. And basically, your sensory is from the receptors, from your sense organs that relay information to your central nervous system. So sensory receptors to central nervous system, that is your sensory division, while your motor or your efferent division relays or commands relay commands from your central nervous system to your effector cells. All right. And again, these effector cells, paulit-ulit lang, are your muscles or your glands. Okay. So some important parts of your peripheral nervous system are your cranial nerves. So these cranial nerves originate from your brain and extend to mostly your head and upper body, while your spinal nerves originate from your spinal cord and extend to parts lower than the head. All right. So your cranial nerves are 12. And basically, yung cranial nerves natin ay the following. Sorry, kukunin ko ang aking kodigo kasi medyo mahirap siyang alalahanin. But let's just enumerate our cranial nerves. O, O, T, T, A, F, A, G, V, A, H. And when you go to your higher zoology subjects, you'd actually find really good mnemonics to remember these cranial nerves. So you have your olfactory, your optic, your oculomotor. Oculomotor, that's how I used to pronounce it, oculomotor. Oculomotor, whatever. Your trochlear, your trigeminal, your abducens, your facial, your auditory, and then you have your glossopharyngeal, vagus, spinal accessory or simply accessory, and then your hypoglossal nerve. Okay, so those are your 12 cranial nerves, and you will be learning more about their function in your higher zoology. And then you have your 31 pairs of spinal nerves. You have your 8 cervical spinal nerves, 12 thoracic, 5 lumbar, 5 sacral, and 1 coccygeal. Coccygeal spinal nerve. Okay. Now here we see this diagram that shows us even more divisions of our peripheral nervous system. So our peripheral nervous system, again, this consists of nerves that carry signals to and from the central nervous system. So again, this can be sensory or motor. And under these, we have your somatic nervous system and your autonomic nervous system. So your somatic nervous system is basically in charge of your voluntary movement, or basically that of your skeletal muscles. And this can actually include some involuntary movement as well. But basically, these are towards your skeletal muscles. While your autonomic nervous system, this carries your nerve signals to your smooth muscles. This controls your smooth muscles and your cardiac muscles, which of course you can find in your heart. So this autonomic nervous system, nerve signals that control your smooth muscles, cardiac muscles, and your glands. Okay, and you can further subdivide your autonomic nervous system into the following. You have your parasympathetic division. So this prepares your body for your rest and digest. All right, your rest and digest phase in which we are conserving energy sa body natin. While in the sympathetic division, this carries nerve signals that prepare for fight or flight response. So parasympathetic, rest and digest, sympathetic, fight or flight. And we have the enteric division, which carries nerve signals that control the muscles and glands of the digestive system. So ito yung enteric division ng autonomic nervous system. And in this illustration, we can actually see the different effects to our body, depending on whether we are resting and digesting or whether we are under the fight or flight response. So again, the parasympathetic division controls your rest and digest, while your sympathetic division controls your fight or flight response. And this figure can show us its effects in the body. Now, in the rest and digest response, what happens in our eye is that our pupils are constricted, while during fight or flight response, our pupils are dilated. Okay? In the rest and digest, there is this simulation of salivary secretion, while in the fight or flight response, the salivary secretion is actually inhibited. So you can actually see all of these effects here. During the rest and digest, our heart is slowed, all right? While at the fight or flight, the heart rate accelerates. Okay, what else? For the bladder, during the rest or digest, this would stimulate urination, while at the fight or flight, this inhibits urination. So if you'd notice, pag nasa fight or flight response ka, hindi ka naihi. Okay, anyway. And then at the rest and digest stage, this will promote the erection of genitalia, while at the fight or flight, this will promote ejaculation and vaginal contractions. All right, so you can just go through that. It's quite interesting to see how our different body parts would respond according to the type of stage we're in, whether we are conserving energy or we are under the fight or flight response. Okay. So finally, this is our last slide, the organization of the nervous system. So just as a summary, again, from the sensory receptors, from our sense organs, comes a sensory input to our sensory neurons, the afferent division of our peripheral nervous system, and these will now be integrated in our central nervous system. And from our central nervous system, commands will be relayed to our motor division in our peripheral nervous system or our afferent division. And this will command in motor output towards our effector cells. And our effector cells, again, these can be your muscles or your glands. So basically, this is just a very simple summary of the nervous system. And with that, that is my last slide. So thank you very much for bearing with me as we talked about the nervous system. And that would be our last one of our last topics for Biology 100.